Okay, let's move that over there. All right, welcome back to Unit 3, Lesson 6. All right, in this lesson, we are going to be investigating how we make P53 and why it is sometimes different, why it's not always the same P53 or the same type. Sorry, there we go. All right, starting Lesson 6 with our learning intention and success criteria. So we're going to be figuring out the DNA codes for proteins, and protein shape affects the function of that protein. Okay, so our DNA, the purpose of it, pretty simply, is just to code and create proteins, and then the shape of the protein that I make determines its function and what it's able to do. All right, success criteria. By the end of the lesson, you should be able to explain how we make P53 and why it's different, just like I said with the lesson question. But now kind of smaller questions within those. What is a gene and where is it located? So what are genes and where do I find them? What do they code for? And what do amino acids code for? And probably, what an amino acid is then. If I change the DNA, what does that do to the proteins? And then what is gene expression? When you hear the word or the term gene expression, what does that mean? Cool. Okay, so we're going to start and you're going to pause the video and talk to your group members, your table mate, someone next to you, whoever is in your vicinity that's on the same lesson, about where P53 comes from and why I have different versions. So why do you, or what do you think where it comes from based on our current understanding from the board game, from all the other things we've looked at with P53, where does it come from? And we've seen that there's multiple different versions of it. What would be advantageous to having multiple different versions? Why don't I just have one version of it? And I want you to think about why you would have different versions of anything, right? Why do I have multiple different copies of like the lesson handouts I provide you guys? Why do you have different shirts? Why do you not just have one shirt, one piece of clothing that you just wash every single day? What would happen to that if I only had one version of this shirt and I had to go home and wash it? Obviously, that would be a lot of work, but then, what is going to happen over time if I only have this and nothing as a backup? Okay, so take a moment, pause the video, discuss those with group members, table mates, and then come back. Okay, coming back, we should have some good ideas about where P53 is coming from, how we make it, and why I don't just have one version of P53. Okay, so now in this lesson, we're going to be doing a kinesthetic model, and it's got a couple of different steps. Kinesthetic means you're actually like moving, right? If I'm doing kinesthetics, that's me moving. So this is just a fancy way of saying you're going to do a model where you get up and move around the room. Okay, so around the room, there's going to be different locations. They'll be labeled like location 1, 2, and on. And at those locations, there's going to be different things. So like at location 1, there's going to be a segment of DNA. So you'll pick up one of the segments of DNA and circle the strand you chose on your handout. And then copy down the complement of the DNA strand. So all of this is going to go in your student activity sheet. So if I click back here on the student activity sheet, you can see go to location 1, circle if I picked up strand A, B, or C. Those are the only 3 options. And then write down the complement here, or type down the complement. And remember that complement does not mean the copy. I'm not saying copy down this exact, copy down the complement of it. And then this note down here is extremely important. So remember to use U instead of T. So for example, if I start with strand A, it would be U, U, C, right? Because the complement of A is T and the complement of G is C, and vice versa. The complement of T is A and the complement of C is G. So if I'm writing down the complementary strand, I would normally do A, T. But like I just said here, if it says T, I switch that with a U. Okay, so rather than T, T, C, it's U, U, C. Okay, and then you're going to continue for all of these, and we chunk them into groups of 3, and that will be important. You'll find later in the lesson. So that's why they're grouped into like little chunks of 3, okay, triplets. So I'll do the second one with you as well for strand A. So you have U, U, C, and then you're going to have G, G, U would be 2. Okay, and then continue using that similar trend Do your best to not make any mistakes, double check it. Try as hard as you can to not make any mistakes. I'm not saying that you won't, but try as hard as you can to not make any mistakes. Okay, so pause the video, go and do that step first, and then you can come back. Okay, coming back, you should have it copied in here, right? That's what this whole thing for. You went to location 1, you copied down the complement of your strand. Now, location 2, edit your copy by crossing out groups 2, 3, 5, and 6. Okay, so I'm going to go back. If I had all of these groups, I'm going to go here and essentially cross them out. So now you can see I'm only copying back in here 1, 4, 7, 8, 9, 10, 11. Okay, so I will not include the copies that I crossed out. Now I know we're doing this digitally, so you can't physically cross them out, so you don't need to, but just in here, you're only going to type in your 1 group, your 4 group, your 7 group, your 9 group, your 10, and your 11 group here. So we're going to create a new complementary strand. Okay, important, don't include the ones that we're quote unquote crossing out. Only include the ones that should be included. All right, step 3, you're going to go to location 2 and find the card that is the complement to each 3-letter combination in your copy. For example, if your copy is UGC, you should look for ACG. So I'm going to go to location 2, and there's going to be a bunch of little cards laid out on the table. Okay, and if on my student activity sheet right here in 1, I have UGC, then I'm going to look for the card that is ACG, or the complement of that. So whatever the complement is. So see how U goes to A, G goes to C, C goes to G, like we just did. You're just going backwards a step, okay? Not literally, but just kind of figuratively right back a step. Okay, when you find that complementary card, write down the code from your card. For example, it could be glycine, arginine, and I don't remember what amino acid that is, but ILE, doesn't matter. Those are just 3-letter abbreviations for different amino acids. So just write down those that are on the back of the card. So you flip the card over, and it's going to have a sequence of 3-letter codes that you write down. Okay, so that will go here. So you're writing down the sequence of the 3-letter code that you had. So if this was GLYARGILE, that is what I would write in this space. Okay, and then do the same for 4, 7, 8, 9, 10, and 11. Okay. Then, finally, you're going to write down the words you find under the code. There is one codon missing from the end. That would be, okay, so there's one missing. The word should be peace. So the last that's missing is peace. So when you can't find 11, the word is peace. Okay. So you're going to find the words that correspond with the code that you had here. So you'll find the words, and you're going to write a sentence. That's what you're doing, right? And we'll see if our sentence makes sense at the end. Is essentially what we're doing here. Okay, cool. So at that point, you should have a sentence written out. It may or may not make sense. It may or may not be revealing. Well, I don't know. We'll see when we get there. Okay. So once you have that, and you've gone through this whole model, now we're going to kind of look back and relate how the component of the model represents the actual living organism, the things that's occurring within my cells. Okay. So starting, what is location 1? Where in my cell am I going through and copying down my DNA into a complementary strand? Where do you think that occurs? Okay, where, for those of you that maybe need some prompting, where is my DNA located, right? It's probably going to occur in a similar place to where it's located. Okay, the paper DNA strand at location 1. What does that represent? And it's in the name of the component of the model. That paper DNA strand at location 1, the actual strand that you're copying down, is my what? And it's in the name, if you're struggling. Okay, the edited copy that I wrote down of that DNA strand. So not the actual strand, but the copy that I recorded. What is that? Okay, and it's very similar to what this was, It's just like a different version of what this was. I can't highlight, sorry, what this one was. Sorry. All right, location 2. Where in my cell is that process actually occurring? When I moved to that second location and went through that process, where do you think that that is occurring within my cell? And it's probably not the same location as location 1, right? Probably not the same part of my cell, but it's probably close by. So if you can figure out where location 1 is, then you can hopefully kind of figure out where location 2 would be, because it would be near location 1. Okay, and then the cards with the codes, what do those represent? So with the codes and the 3-letter abbreviations of those different things, what do those represent? Right, those are kind of like the building blocks of my larger thing, right? I put those together to get my larger molecule, for lack of a better word, my sentence, which was essentially my molecule of some kind. And then what was the sentence? That whole molecule together we call what? Okay. And I know I'm being pretty vague here in the video, so if you're struggling with those things, because it can be a little bit difficult, work with your table partners, work with your group members. If you need my assistance on those, I can help you a little bit more. But just kind of think of like, what am I going through and doing, and where would those occur within my cell, and what is the end product that I'm making as a cell, not as a student. I know what you made; those are all here, but what do those represent in the cell? Okay, so now we're gonna go through our making sense discussion. So we will also have this in the small groups when we're there, but you're going to record answers kind of on your own, or not on your own, with your group members. Let's work through it with your group members, and we'll talk through it with me. But you're going to start with how the model helps you answer the question, how we make P53 and why it is different. So to start, we probably need to know what P53 is, right? And if hopefully we already have a pretty good understanding at this point coming in that P53 is a protein. Okay. And so the process that we just modeled, what was the end goal? What did we make at the end of that process? What was the sentence, right? And that should help me understand what P53 is, and then why it's different. So like, what would occur in that process that you just modeled in that kinesthetic model that could make P53 different sometimes? Okay. Okay, so then find someone that worked on the same DNA strand as you. So it needs to be someone that worked on strand A if you did strand A, or worked on strand B if you did strand B, or whatever strand you did needs to be the same. Otherwise, this doesn't work, right? Don't use someone different, or your sentences will be different because your strands were different. Anyway. And you're going to compare the sequence of amino acids and words and see how they compare. If they are different, why do you think they are different? So you're going to find someone, again, with the same strand as you, and they should have the exact same amino acid sequence, those 3-letter abbreviations put together, and the same sentence at the end, that same end product. But they might not. And if they don't, why would they be different? What do you think could have occurred in this process, just naturally, to no one's fault, it's no one's fault, that would make them different? Okay. And just think about, like, as you were going through it, what were you thinking, right? You were probably all thinking like, oh, this is difficult, right? I'm kind of looking back and forth. These are a lot of letters. There's a lot of opportunity for me to maybe not copy down correctly. So what would that create down the line if I didn't copy it down correctly, and maybe didn't write down the complement correctly or something like that? All right, and then find students that worked on the 2 different strands now. So if I did A, I need to find a student that did B and a student that did C, and compare their amino acid sequence and words to mine. Now, these probably shouldn't be the same, right? Because we had a different DNA strand. So our end product is probably going to be different. So we're going to look and see, are they different? And if they are, why do you think they're different? Why are they not all exactly the same? And you'll probably, like, go all the way back to the DNA strands that you guys had, and were they all the same? Okay. And if they weren't all the same, does it make sense that the end product's not all the same? If they were all the same, then maybe it's a little bit more confusing why the end products are all different. And maybe all the end products are the same. I don't know. I'm just, if they are different or if they're the same, how did that occur? So it could go the other way. It could be they're all the same, but have different DNA. And how do you think that's possible? So any of these are options. I'm not saying definitively what really occurs. You'll have to go through the model to figure that out. But when you observe that, why do you think that you observe differences or similarities in those models when we know the DNA strands were not the same? Okay. So now some of this might be a little confusing, right? Some of these aspects might not have been very intuitive to you, and that's okay. That's why I've been kind of vague, is I've got a plan for that down the line here, where you at this point, you've gone through all of the rest of this, right? You've done this, you've done the making sense questions, you're right here on the reading P53 and DNA. I'm going to click on the link, and it's going to take you to this reading, okay? Again, it's super short. All the readings in this unit have been really short, okay? So, like, don't tell me it's too much reading, because it's a page, not even front and back. It's like literally 4 paragraphs. So go ahead and read through this, and you're going to read more about cells and DNA and the process of making proteins. Okay, so what does my DNA do? What does that DNA strand make? If you were struggling with that, now I've pretty much already told you, and it tells you what P53 is, and kind of a little bit more about amino acids and some different very important molecules that we maybe don't know the name of yet, but you will after this reading. So this is going to shed a lot of light on that, comparing the model to the actual cells if you struggled with that, and those making sense questions. So after reading this, should be very easy to go back and maybe revise some answers that you had before. Okay, so this is going to provide a lot better and clearer understanding, deeper understanding of those making sense questions. And then also just record anything you notice and wonder, right? If there's something that you find interesting that wasn't included somewhere else, put that down. If there's something that you didn't know before, please record that. And if there's questions that you have about this whole process, about DNA, about chromosomes, about P53, then go ahead and record them there. Okay, cool. So we read that reading. Please don't ever skip the readings. You will miss just so much key information by skipping the readings, okay? Okay, so we're going to build some understanding here. This as well, I will talk with you in your small groups, but you should work on it together in your small groups first, and then just wait for me to come kind of provide some expertise on it. So we figured out really big ideas about the process of making proteins from the coded DNA. That process is called gene expression. Okay, so some things we need to think about. My prompts, you're going to record answers in this big space, so that it's not really a super like formatted part of the document. Is just a big empty space for you to record your thinking as you guys discuss and as you discuss with me. Okay. So why did we have different versions of the DNA? Why did I not have identical copies of the DNA? Okay. And again, thinking why, as a human being or as an organism, would I want multiple different copies of the DNA? Why would I not want all of my DNA to be exactly the same and to code for one thing, one protein? Why is that not going to be advantageous for me to code for a single protein? And then what did each version code for? Which I kind of told you, right? They all code for proteins, but did they all code for the same one? And why, again, is that important for me as an organism that they do or don't code for the same thing? Okay, how did the structure of each of the different versions of P53 affect the function? Okay, and again, if you read the sentences, I will tell you that. So how did having a different sequence of DNA and a different amino acid sequence, which made a different structure of my sentence or my protein, how did that affect how well my P53 worked? Okay, what happened in places where the sentence did not match the key? What did that represent in the cell? So if I had a sentence that did not match the key that I gave you of what those sentences should have been, what does that represent? And does that, occur naturally in my cells? And when it does, what would we call that? Okay. And then what do you think the shape of P53 protein would look like in a case where the sentence did not match the key? So if the sentence, P53 is the sentence, right? And my sentence that represents the P53 protein does not match what it should be, how is that going to impact the function of my P53? So if my P53 is not structured appropriately, like could have happened to the sentence, what is going to happen to the function of the P53? Is it going to function as well as it should? Is it going to continue to do its job as well as it should, or not? Okay. All right. And by the end of that discussion, all of those big ideas, we should be ready to fill in our progress tracker answering these questions. Again, reminder, you do need to record like answers to all of these. These are the same as your success criteria. So you do need to tell me what a gene is, where they're located, what they code for, what amino acids code for, if I change the DNA, what it does to the protein, and what the process of gene expression actually is. So that way, if I or anyone else ever asked you what gene expression is, you would be able to explain what gene expression is. Okay, and then these are the questions we're going to look at moving forward, more about mutations accumulating over time and other things that could cause cancer. Okay. If you have any questions, if you need anything working through this lesson, please let me know and have a great day.